It's the biggest mass extinction since the dinosaurs disappeared. Every year, countless species are lost. But is that permanent?
We're going to have to dip into the past and bring some of these really important species back.
It is hoped that technology will enable this.
The rhinos need our help, and I want to help them. I can help them.
The aim is to give new life to the genetic material of dead animals.
Who is financing this? What are their motives, and isn't the money needed elsewhere?
Things are progressing rapidly in laboratories around the world.
Our goal is to have our first mammoth calves by 2028.
We are opening a Pandora's box and have no idea what to expect.
This is Najin and her daughter Fartu, two northern white rhinos, the last of their kind. The species has been almost exterminated to harvest their horns. The two females are guarded around the clock at a fenced-in sanctuary in Kenya. In the wild, only southern white rhinos are left. They are related but have different language and genetics. The last northern rhino bull died in 2018, so there can be no more offspring. When Najin and Fartu die, an entire species will become extinct. Nothing more can be done. Or can it? Experts in Berlin are not yet admitting defeat. The taxpayer-funded Leibniz Institute for Zoo and Wildlife Research leads the international scientific consortium BioRescue. Reproductive medicine specialist Thomas Hildebrand has been the head of this project since 2019, which aims to prevent the extinction of the northern white rhino.
When you work to save such a large mammal, you are faced with almost unsolvable problems and have to develop completely new approaches.
The Cryo Laboratory. For decades, researchers have been storing tissue samples here, including egg and sperm cells from 300 endangered animal species. It's like a frozen zoo. The cells are preserved in liquid nitrogen in a kind of hibernation state at -193 degrees and thawed as required. They include sperm from 4 northern white rhino bulls that died in zoos. These samples are crucial for Thomas Hildebrand and Susanne Holze. Eggs have been retrieved from Fartu and Najin. These will then be fertilized in the laboratory with the preserved sperm of the dead rhino bulls. An embryo develops in nutrient fluid and is implanted in a cow of the related southern white rhinoceros. In the womb of the surrogate mother, the embryo develops into a rhinoceros calf and is born after 16 months. As soon as possible, the little one will then be placed with Fartu or Najin to be brought up. It is an experiment on wild animals that have a special significance.
The rhinoceros is a keystone species, which means that it's at the top of a hierarchy of entire species communities. Repairing an ecosystem by bringing back a keystone species is a very important element in repairing the planet.
Hildebrand's mission began in 2019 in Kenya. At the Olpajeta Game Reserve, the vet prepares the operations. It's a race against time. The calf has to learn the behavior of its last 2 living species members before they die. The two females can no longer bear offspring themselves. Fatu, born in 1989, has cysts and inflammation in her uterus, and her mother Najin, who is 11 years older than her, has injured hind legs. However, both are still producing healthy eggs.
Right now, how she looks, I think you did a very good job to keep her healthy. With a mobile lab we bought, so we will analyze her blood and then check the blood parameter.
Hundreds of kilos of equipment have been flown in. The experts have gained a lot of experience through similar procedures on zoo animals. But they do not know how a wild animal will react to the removal of eggs. Fatu is under anesthetic. Her heart rate and breathing are being monitored. The procedure being performed by Hildebrand's team is risky for the animal, since the sexual tract of a female rhinoceros is like a labyrinth. An ultrasound probe helps to monitor the operation. The specimens look healthy. The female rhino is unharmed and recovers quickly. Her eggs take a long journey to Europe, to a special laboratory in Italy. White rhinos still have habitats and could survive if they were not hunted. According to studies, human influence means that species die out hundreds of times faster than they otherwise would. There have been at least 5 major species extinction events in the history of the Earth. About 450 million years ago, the sea level dropped and the planet became bitterly cold. 85% of species died, after which many new ones developed. The next event was massive volcanic eruptions and oxygen depletion in the oceans that killed off almost all life. An asteroid impact triggered the next disaster. A good 60 million years ago, a meteorite crashed into the Earth, followed by earthquakes and tsunamis. The sky darkened and the planet became extremely cold. Almost all dinosaurs disappeared in this event, along with many other species. Can we bring back dinosaurs like in Jurassic Park? Probably not, because their genetic material has decayed over time. But the last mammoth died only around 4,000 years ago. Is there any chance of reviving this species without using preserved cells as with the rhinoceroses? In addition to the deterioration of their living conditions, hunting by humans also played a role in the extinction of these ancient elephants. All that exists now are finds in the frozen ice of Russia and Alaska. The idea of resurrecting them has been discussed for decades. For a long time, however, there was a lack of sufficient genetic material and methods to replicate the genes. This is now changing. Thawing ground in Siberia has repeatedly revealed well-preserved specimens. This has made it possible to reconstruct completely the mammoth's genetic material from tissue, bones, and blood. In Dallas, in the US, a private company wants to take the next steps. Colossal Biosciences has been working on de-extinction projects, i.e. the revival of extinct species, since 2021. More than 60 scientists are involved in this research. $225 million of company capital comes from investors, including a company financed by the CIA. Colossal Biosciences states on its website that it is committed to restoring the Earth to a healthier state. They want to revolutionize history and be the first company to successfully apply modern genetic engineering to extinct species. The brains behind this project are scientist George Church and entrepreneur Ben Lamb.
We saw an opportunity to, one, bring back iconic species that humans had a role in their extinction and kind of undo the sins of the past, rewild them back into the ecosystem in partnerships with like indigenous people groups and private landowners and governments as a way to help restore ecosystems. And on the path to that, we want to build technologies that help modern conservation that can be applied today.
Colossal's flagship project is the mammoths. Their closest relatives are Asian elephants, whose population is declining because their habitats are shrinking. The idea is to open up new areas in colder regions for this species thanks to mammoth genetic material.
The only reason we're looking at ancient DNA at all in this case, under that framing at least, is because those elephants were exceptionally cold resistant. They could live for months at minus 40 degrees. So that's a rare resource that we want to exploit.
The genetic material of the Asian elephant is 99.6% identical to that of the mammoth. The remaining 0.4% will be covered by a new type of technology. Certain gene segments are removed from the genetic material of a fertilized egg cell of an Asian elephant cow using CRISPR gene scissors. With the artificially recreated DNA, the blueprint of which originates from mammoths, genes for woolly hair and a thick layer of fat can be precisely implanted into the genetic material of the elephant cell. Via electrical stimulation, the manipulated egg cell grows into an embryo. It will be planted in the womb of a surrogate elephant mother. The offspring will then have the cold resistance of a woolly mammoth and will probably look like one too. Mammoths and elephants carry their young for 22 months. It would take a long time to produce these hybrid creatures, and it would take many surrogate mothers, maybe too many.
We're working for a variety of reasons on developments outside of the elephant's body, from embryo to newborn, ex vivo and artificial wombs and that sort of thing.
Is this possible? In 2017, a team of American researchers managed to grow premature lambs in a biobag, a type of plastic bag. Since then, many scientists have been working on developing artificial wombs.
Then we'll establish centers throughout the Arctic where they will be born, right near where they're going to be. So you don't need lorries. They walk out the door of the lab and they're where they belong.
And this is where these mammoth elephants will graze. In Siberia's former mammoth steppe, where grazing animals are scarce and forests are now growing. In 2018, George Church met Russian biologist Sergei Zimov, who has been running a special research station there for decades, an ice age park. He has settled bison, musk oxen, and wild horses over a large area. Why? The herbivores scrape away the snow to find food. They remove the insulating layer of snow on the ground. Cold air reaches the soil and ensures that it remains frozen and does not thaw. This means that less stored carbon dioxide is released from the soil into the environment. Mammoths could therefore help to protect the climate. However, this would require thousands of Colossal Biosciences laboratory animals. Would the mammoth colonies be good for the natural world? At the Senckenberg Natural History Museum in Frankfurt, Katrin Böning-Gese researches biodiversity, i.e. the relationships between animals, plants, and their habitats.
That would be a really wild experiment. The mammoth has parasites and has an impact on the vegetation. There are competitive relationships with other species. It's as if we'd been threading the colossus into a complex network. And it affects all the other threads, so we can't predict in advance what will happen.
The return of the quagga is being worked on in South Africa. There are also plans to recreate the American passenger pigeon. There are no known plans for the giant auk. The situation is different for the flightless dodo. Researchers at Colossal want to bring back the bird that was wiped out by human-introduced rats.
Bringing extinct species back to life is obviously something that fascinates people and is understandable. We can save and bring back what we have lost. But in terms of protecting biodiversity, it's a tiny drop in an ocean of problems.
According to studies, up to 58,000 species become extinct every year. Their genetics are lost and they leave gaps in the network of relationships in their habitat.
The more species we have, the more robust and stable ecosystems are. This means that every time we lose a species, we weaken this network of life, and this creates uncertainty. And all the ecosystem functions that we obtain from nature are becoming increasingly difficult to grasp and produce.
The rescue of the rhinos continues in northern Italy near the city of Cremona. The Biorescue Research Consortium includes Avantea, a leading specialist laboratory for horse breeding. Experts are working on combining the genetic material of highly productive farm animals. Since horses are related to rhinoceroses, much is also similar in their embryonic development. This is why all of the northern white rhino eggs from Kenya are brought here to be fertilized. But the researchers are breaking new ground with these wild animals.
We have to discover the very specific requirements that the rhinoceros embryo has and identify these in long series of tests. Some egg cells don't tolerate fructose, some glucose, and you have to find that out. That's a lot of small things, but they have a significant impact.
Compared to horses, the number of eggs obtained from the two rhinos is small, and not all of them survive transportation and the procedure. Before they are fertilized, the embryonic germ cells still have to mature at a constant temperature. Embryos are produced here not only from northern white rhinos, but also from the more common southern species. The researchers want to practice the embryo transfer method on these animals first. Thawed sperm from deceased rhinoceros bulls are injected into the egg cells. The eggs then divide regularly in the incubator until they are sufficiently multicellular. Their growth is interrupted after 8 days. All embryos are then stored in the cold chamber for later use in Kenya. In the Ol Pejeta sanctuary, embryo use is first being tested on the southern white rhino. This is where Mother Nature comes in. As well as the surrogate mothers, a bull has also been selected. His presence stimulates the females and indicates whether one of the partners is ready to conceive. The bull has been sterilized, as he is not supposed to father any offspring with a surrogate mother. If mating occurs, the researchers only have 6 days to implant the embryo created in the laboratory into the cow. On September 23rd, the biorescue researchers get ready. The vet has to get close to the female with the anesthetic gun. This is because the range of the dart, which is fired using air pressure, is short. The anesthetized rhinoceros cow is immediately protected from overheating. Hildebrandt and Holze have previously tested 13 embryo transfers in rhinos in zoos and on farms, each time unsuccessfully. They have developed all the methods and technical equipment for this species themselves. With this needle system, Hildebrandt has to be careful not to hit any of the large blood vessels when inserting the embryos. The rhino survived the procedure well. From now on, it's a case of waiting and seeing. In Kenya, Berlin, and Cremona, the Biorescue Consortium is working to save the rhino. Support is now also coming from Dallas. The private consortium, Colossal, is not only working on reviving the mammoth, it is also seeking partnerships with other scientists. It has been cooperating with the Berlin Institute for Zoo and Wildlife Research since 2023.
It has to be said that companies like Colossal work closely with us and provide us with technologies developed for our needs. This means that in addition to their efforts to bring back the mammoth, they are helping us in our conservation efforts to use these highly complex technologies for the northern white rhino.
The American company is particularly interested in the experience gained in reproductive biology.
You have to have all of the top experts, you got to have all the specialists, you have to have a bunch of technologies, then you need a lot of infrastructure. So, we raised $225 million to go support these initiatives. And then some of the technologies that we developed that have an application to human healthcare, we're monetizing for economic returns.
Colossal itself is a colossus among high-tech companies. The biotech company was valued in 2023 at $1 billion. In the German university town of Tübingen, a biologist is investigating the effects of the work involved in reviving extinct species. As an independent environmental ethicist, Uta Esa advises authorities and universities. Should we spend a lot of money on mammoth projects?
The fact that someone earns money with something is not a bad thing per se, but of course the question is who is financing this and what are their motives? And might the money not be needed much more urgently elsewhere?
Until now, environmental organizations have spent a lot on preserving protected areas. The modern approach of investing large sums in the return of extinct animals might not always aid the rather conservative approach of preserving living species.
There is a risk that in the hope that we can simply rebuild everything again, the efforts that would have to be made to preserve biodiversity now will decline, that we won't put enough human and financial resources into it and say, well, once they're extinct, we'll just rebuild them again.
The concern is, if you can repair like this, then you won't have to worry about it at all, because you can repair all of humanity's mistakes. I don't think that will be the case, because it's simply far too expensive. It's an extreme expense. The commitment in terms of scientific technology of ours, it should be the exception.
Even now, the rescue efforts for the rhinoceros would hardly be feasible without funding from German taxpayers. Funding for nature conservation is limited.
We're not taking capital away from conservation. This is new money going into conservation. And all of the tools and technologies that we make on the path to de-extinction, we're giving to the world, to governments, to nonprofits, other NGOs for free. So any nonprofit can use our technologies for species preservation and helping save critically endangered or other endangered species.
Colossal has also been supporting researchers in Australia since 2023. The focus there is on an animal with stripes like a tiger and a head similar to a wolf. It was once the largest carnivorous marsupial on the continent. With a set of teeth to prey on kangaroos and wild rabbits, the thylacine is also known as the Tasmanian tiger. This film footage shows the last living specimen in a zoo. After sheep were introduced to Tasmania, farmers saw Tasmanian tigers as enemies and hunted them mercilessly. On September 7th, 1936, this animal died, and with it, its species. The remains of animals from zoos are stored at the Museums Victoria in Melbourne. Curator Kevin Rowe is interested in them.
Tasmanian tigers are a symbol of extinction and loss in Australia, and as we know, Australia has the worst extinction record for mammals of any country on Earth, with 30 species lost since European settlement in Australia. The Tasmanian tiger is gone. It was an important predator on the Australian landscape, and if we care about the functioning of our ecosystems, we need to sustain the top predators in those systems.
This man wants to help. Geneticist Andrew Pask from the University of Melbourne. He wants to revive the thylacine. Others have had the same idea in the past, but Pask is now being supported by Colossal with millions of dollars and has newly developed technologies at his disposal. The scientists are sifting through the largest collection of thylacines in the world. There are specimens of 32 of these animals in the Melbourne Museum.
Yeah, there's some good tissue there. We're hoping that from some of these specimens that we've looked at that there's some really nice intact DNA there and that we can sequence that entire genome of these animals. And that really gives us the blueprint or the building blocks of what we need to recreate and bring these animals back.
Hope. Embryos preserved in alcohol, of which there are only 13 worldwide, 3 of them in Melbourne.
Yeah, it's remarkably well preserved.
Andrew Pask also takes samples from these specimens.
Fantastic. Oh, that's amazing.
The embryo labeled C5757 is over 100 years old and comes from the womb of a female preserved in the museum. It is well preserved and therefore a promising candidate for Pask's plans.
We still can't create life where there is none. So the way that de-extinction works is you have to begin with life. So what you do is you have to be able to get all of the DNA sequence of your extinct animal. And then once you have that, you compare it to all living animals to find what is the closest living relative of the extinct species.
Embryo C5757 is the breakthrough. In 2018, the blueprint of the Tasmanian tiger was almost completely reconstructed for the first time. Now Andrew Pask and his team begin their search for a genetically closely related animal. Researchers at the University of Melbourne have succeeded in finding a close relative of the Tasmanian tiger. The fat-tailed, narrow-footed marsupial mouse, or dunnart, which shares ancestry with the Tasmanian tiger. A comparison of its genetic material with the reconstructed DNA reveals an astonishingly close relationship to the large predator. The laboratory mice have many advantages for the project. The small animals are relatively common in the wild and have a short gestation period. They reproduce well and are easy to keep. This is doubly valuable for research. Special thylacine genes can be introduced into the cells of the dunnart using gene scissors. And the finger-long carnivores are also suitable as surrogate mothers that carry the embryos of the Tasmanian tiger.
There's obviously a massive size difference in a dunnart to a Tasmanian tiger, but this is one of the fantastic things about marsupials is when they're born, they're all really, really small. So that means that even a dunnart could gestate and give birth to a baby Tasmanian tiger.
Whether dunnart, koala, or kangaroo, the embryos of all marsupials are about the same size in the womb, usually barely bigger than a grain of rice. They all give birth to their offspring at a very early stage of development. After the gestation period, the young crawl out of the birth opening. In the mother's abdominal fur, they shimmy to the pouch, climb in, and attach themselves by the mouth to one of her teats. Well protected, the young animals mature. It was the same with the thylacine. But which genes make a Tasmanian tiger a Tasmanian tiger? Pictures of the skeleton and organs have been taken of all of the embryos. PASC concentrates on typical characteristics: the snout, the jaw, the strong hind legs. In comparable predators, such as the dog-like dingo, he finds gene regions that are important for hunting prey. And these could also be decisive for the reconstruction of the Tasmanian tiger's DNA. The researchers are studying which introduced Tasmanian tiger genes trigger which changes using embryos manipulated in the laboratory that came originally from dunnarts. There are still many unanswered questions, but knowledge about the various stages of development is growing rapidly. The first Tasmanian tigers could be born in the next decade.
If you make all of those changes, at the end you would have a complete thylacine, and any differences that you would have from any wild thylacine would be about the same that any two animals in the thylacine population would have from each other, or you and I would have from each other. So at that point, I think you genuinely have a thylacine that you then could put back into that ecosystem.
It is not yet known whether the genetic makeup of the thylacines will be sufficient to enable them to survive in the wild. Should reconstructed animals be released back into the wild at all?
God is, so to speak, the decision maker as to who may live and who must die. This is not a decision that people make, but one that they are overwhelmed by, and biopolitics can now decide who can live and who must die.
We manipulate the landscape, we're playing God. When we drove this animal to extinction in the first place, we were certainly playing God then. And so I think what we're doing now is trying to correct some of those mistakes we made in the past.
That would be extremely arrogant for someone to claim that we're just playing. We're not even playing. We're working at being engineers. It's what humans have been doing since we became humans. We study the past. We plan for the future. We invent things. So no, we're just doing our job. That's all.
Attitudes such as esteem, reverence, respect, and humility are completely lost. It's the complete opposite. It's arrogant to say we can make what nature has produced ourselves, and no longer seeing that perhaps we, as part of the whole, are not in control of things the way we would always like to be.
So is it the case that we are not only making corrections, but we're also opening a Pandora's box?
Wir spielen auf keinen Fall Gott, wir machen nichts Neues, wir kreieren nicht neue Formen des Lebens, sondern wir nehmen das, was von diesen beeindruckenden Tieren in Museen, in Kryolagern und in Form von lebendem Material vorhanden ist, bringen das wieder zusammen und machen daraus neue Babys.
Hildebrandt is also working with the Max Stellbrück Center in Berlin to generate test tube rhinoceroses. Research into stem cells is being carried out here. To create healthy egg cells, Vera Civica has grown cultures of skin tissue from 12 northern white rhinos that died in zoos. Living cells from tissue or blood are removed from a rhinoceros and preserved. They can be returned to an embryonic stage at any time using a cocktail of genes. These stem cells are then all-rounder cells from which any body cells can be cultivated. If certain starter proteins are added, the stem cells mature into sperm or egg cells in a special culture medium. Once researchers have successfully produced the mature egg cell from the skin cells, this is fertilized with frozen sperm from deceased males. The resulting embryo can be implanted in a surrogate mother, the related southern white rhinoceros, and the calf she is carrying. The advantage is that the artificially produced egg cells can be used to increase the genetic diversity of the future rhino community. The researchers would no longer have to rely solely on the gametes of the two closely related females. However, they must ensure that all the aids used do not alter the genetic material of the egg cells produced.
When I have made a stem cell, I carry out very thorough quality control to make sure that there is nothing in there that doesn't belong. After all, I want to have a healthy rhino at some point, and not create something genetically modified. That is not the aim of my work.
So far, it has only been possible to produce egg cells precursors. They only mature when they are surrounded by ovarian tissue. However, it is not possible to obtain this from living or deceased rhinos. The nourishing cell material must therefore also first be cultivated.
If we have to use this method, it's already too late to do anything else. Then all other traditional species conservation measures have failed. It's simply the last chance to save this species. And we see it as a blueprint for other species that are similarly endangered.
Time is of the essence because almost all rhino species are endangered.
In the long term, a whole herd could be re-established this way.
Good news from Kenya. The surrogate mother of the southern white rhino is pregnant, and the embryo has been growing for 2 months. In December 2023, however, heavy rain sets in. As a result, bacteria from the soil are flushed to the surface. The surrogate mother dies of poisoning, and with her the embryo. The researchers from Berlin are left with only the dead fetus. The artificially created embryo lived in the womb for 62 days. It was male and already 6.4 centimeters long. The tissue samples, as well as ultrasound examinations, revealed that he was perfectly healthy. There was a 95% chance that the baby would have been born after a total of 16 months.
It was a really mixed feeling because we had never such beautiful little rhino in our hand. But on the other side, we would strongly prefer that it is still in the uterus and growing.
The little one's surrogate mother was also healthy until her bacterial infection. The procedure had not harmed the mother.
This little male fetus I hold in my hand was a really emotional moment, but it also represents a dramatic scientifically breakthrough. You can see it here on
the successful effort will be presented at an international press conference in January 2024, including a 3D model.
This is our baby. It has the name number 1.
Now the team wants to implant a northern white rhino embryo into a surrogate mother.
That is really a milestone to allow us to produce northern white rhino calves in the next 2.5 years.
The success of the international biorescue team is exemplary. Top researchers from all over the world are working on scientific benchmarks for future species conservation.
All of the tools that you need to do de-extinction, to bring an animal back, exist today. So there's no magic, there's no technologies that we need to develop in order to make this a reality.
The methods are getting better and better, and a lot can be achieved with a lot of money.
I think, there's a lot of extinct species that could be very adaptive in the modern world. You just want to be very cautious that you don't create an invasive species.
It remains controversial whether there should be any new old species at all.
1 million species threatened with extinction. We know exactly what needs to be done to save these species. And going in there now with such technological solutions is well-intentioned and fascinating, but not really helpful.
Whether biotechnology can now save nature remains controversial.
I can't see this as a contribution to nature conservation. That would rather be to address the drivers of the destruction of nature than reconstructing individual species.
Or will it make sense to preserve nature and reconstruct animals in the future?
We assume that we will repair what has broken in the past and hopefully not have to repair what will break in the future.
Even if bioengineers were able to produce northern white rhino calves again, these animals need intact habitats. Should other extinct species also return? The debate will only really get going if and when the first mammoths and wolves are actually born.